Welcome to SS2 Agricultural Science, Term 3, Week 2. Tissue culture, also called micropropagation or plant tissue culture, is a laboratory technique for growing plants from tiny pieces of tissue in sterile conditions. It is one of the most powerful biotechnological tools available to modern agriculture. By the end of this lesson, you will define plant tissue culture and state the underlying principle, totipotency, describe the process of tissue culture, list the advantages and applications, explain the limitations and challenges, and answer WAEC agricultural science examination questions. Let us begin. Totipotency, the ability of a single plant cell or small piece of plant tissue to grow and develop into a complete plant. This is the biological principle underlying plant tissue culture. Every plant cell contains the complete genetic information, DNA, for the whole plant in its nucleus, the same DNA as the original plant. Under the right conditions, hormones, nutrients, light, temperature, a single cell or a small piece of tissue can be induced to divide, differentiate, and eventually regenerate a complete plant. This does not occur naturally under normal conditions. Cells become specialized, but it can be triggered in the laboratory using plant growth regulators. Key plant growth regulators, hormones, used in tissue culture, auxins, e.g. IA, IBA, NA promote root formation, at high concentration, suppress shoot growth. Cytokinins, e.g. BAP, kinetin promote shoot growth and cell division, encourage multiple shoot formation. The ratio of auxins to cytokinins determines development, high auxin, cytokinin root formation, low auxin, cytokinin shoot formation, roughly equal callus formation, undifferentiated cell mass. Culture medium, a sterile nutrient jelly, agar, or liquid containing minerals, murashage and skug, WMS, medium is standard, sugars, sucrose as carbon source, since plants cannot photosynthesize in culture, vitamins and amino acids, plant growth regulators. Stage 1, explant selection, choose a small piece of plant tissue, or the explant, typically a shoot tip, node, leaf section, root section, or embryo from a healthy, disease-free mother plant. Stage 2, surface sterilization, the explant must be free of microorganisms, bacteria and fungi, that would overgrow and kill the plant tissue in the nutrient-rich culture medium. Sterilization, wash with 70% ethanol, alcohol, for a few seconds, soak in sodium hypochlorite, household bleach, 10 to 20%, for 10 to 20 minutes, rinse several times with sterile distilled water. All subsequent operations must be carried out in a laminar airflow LF, cabinet, a sterile workbench with air filtered through HEPA filters. Stage 3, initiation culture. Place the sterilized explant onto the initiation medium in a sealed, sterile culture vessel, usually a glass jar or tube. Incubate at controlled temperature, 25 to 28 degrees Celsius, with appropriate light conditions, 16-hour photoperiod or in the dark depending on the stage. Stage 4, multiplication, subculturing after 4 to 6 weeks, shoots, or callus, develop, transfer, subculture, to fresh medium to allow continued multiplication. Each subculture potentially multiplies the number of shoots by 3 to 10 times. This can be repeated many times, generating thousands of plants from one explant in weeks. Stage 5, rooting, transfer shoots to a rooting medium, high auxin, cytokinin ratio, to induce roots. Stage 6, acclimatization, hardening of transfer the rooted plantlets from the sterile agar to soil or compost in high humidity conditions, a mist propagation unit or cover tray, gradually reduce humidity over 2 to 4 weeks. The plantlets adapt from a nutrient-supplied, low-transpiration environment to an independent, soil-growing condition. Applications of plant tissue culture, rapid mass propagation, from one disease-free mother plant, thousands or millions of genetically identical plants can be produced in weeks. Example, banana plants, plantain, tissue culture plantlets can be produced to supply farmers nationwide much faster than traditional suckers. Oil palm, cocoa, rubber, pine, eucalyptus, all propagated by tissue culture for commercial forestry and plantation agriculture. Disease elimination, meristem tip culture, using the very tip of growing shoots, which is often virus-free even in infected plants, can eliminate viruses from clonal material, producing certified virus-free planting material for potato, sugarcane, strawberry. In Nigeria, cassava, yam, and banana are routinely cleaned of viral diseases using meristem culture by IITA and NERDC-approved. Germplasm conservation, storage of genetically valuable plant material in small volumes at low temperature, cryopreservation, preserves genetic diversity. Endangered plant species can be conserved and multiplied. Production of specific compounds, plant cells can be cultured in liquid medium to produce secondary metabolites, alkaloids, flavonoids, taxol, and anti-cancer compound from Utri cells. Genetic transformation, tissue culture is the entry point for creating genetically modified organisms, GMOs, introducing foreign genes into plant cells and then regenerating whole plants. Limitations and challenges of plant tissue culture: high cost, requires laboratory equipment, LAF cabinets, autoclave for sterilization, controlled environment chambers, glassware, and training, not accessible to small farmers. Somoclonal variation, plants produced through callus culture may not be genetically identical to the parent due to mutations during the undifferentiated callus stage. Shoot tip culture minimizes this risk. Acclimatization losses, tissue culture plantlets have thin cuticles and low epicuticular wax, they wilt easily during transfer to soil, careful hardening off is critical. Skilled labor requirement, contamination of cultures through lapses in sterile technique can destroy entire batches. Limited to vegetative propagation characteristics, quality of offspring depends on genetic material of mother plant, cannot improve upon the parent genotype, unlike hybridization. Relevance to Nigeria, IITA, a body on world-leading research on cassava, yam, maize using tissue culture. NERDC-approved, National Root Crops Research Institute, UMIDIC produces certified disease-free yam and cassava seed material. NABDA, National Biotechnology Development Agency promotes biotechnology including tissue culture nationwide. WAEC exam questions, define tissue culture, state the principle, totipitancy, list advantages and limitations, describe the steps in tissue culture, name crops produced via tissue culture in Nigeria, compare tissue culture with conventional propagation. Key points: tissue culture, growing plants from cells/tissue in sterile nutrient medium in a laboratory. Totipitancy, every plant cell has the potential to develop into a complete plant, has complete DNA. Steps: explant selection, surface sterilization, initiation, culture multiplication, subculturing, rooting, acclimatization. Key requirements: sterile conditions, LAF cabinet, autoclave, OMS nutrient medium, plant growth regulators, auxins for roots, cytokinins for shoots, controlled temperature and light. Advantages: rapid mass propagation, disease elimination, genetic conservation, year-round production, small space requirement. Limitations: high cost, somoclonal variation, especially from callus, skilled labor, acclimatization losses. Nigerian crops: cassava, yam, banana/plantain, oil palm, cocoa, tissue culture used by IITA, NERDC-approved, universities. WAEC test, define, state principle, describe steps, advantages/limitations, crops produced, comparison with conventional propagation.